Hi everyone, it's a bit intimidating to be talking in front of you all, but I guess I have some things to say. I didn't start as a data scientist. In fact, I started with a master's in biology. I wanted to do something with my hands. It didn't work, so I went to analyze the data made by others. This worked a bit better. So we are all data. I don't know if you're aware of that, but you are massive amount of data. In your cells, the DNA is made of only 4 components, which is A, T, C, and G. It's just the 4 small bases that make all life on Earth so far, as we know. In your own body, there are about 3 billion cells. Oh, well, no, your genome is made of 3 billion base pair, so this is more or less the same for everyone. But your body is made of 4 to the 10 to the power 13 human cells, and you have about twice that much bacteria that are living in your gut. So it's huge amount of bacteria, huge amount of cells, and it's even less amount of data that the humanitarian interaction that we're recording every day on the internet. So whatever you are doing on the internet, it's being recorded, it's generating new data. So given the small amount of, well, information that you are just generating chaos everywhere. So my talk will be on 3 parts. It will be basically chronological. I started my PhD during biology. Then I did things for Kaggle, and I wanted to see how I compared in data science. And then I went on to marketing, and this will be the conclusion of my talk. I like to have this illustration from Hitchhiker's Guide to the Galaxy. I don't know if you know it. They built a huge computer, asked it what was the meaning of life and universe and everything, and the answer was 42. So this is what you get when you don't know what the question is, and you just feed data to a computer. Why is it interesting to deal with biology? I don't know if some of you work in this field, but it's one of the richest and most intensive field in data science because it's very tricky to deal with it. As we said, we have a huge amount of data because, well, we have cells, we have DNA, we have many information, but unfortunately there is only a very limited number of people on Earth, and even more, only a fraction of them are ill, which is nice, because we don't want everyone to be ill. That would be unethical. But still, if you want to make tests and to understand which is the best treatment that you want to give to a patient, you want to have at least a large amount of data. And this is where is the needle in the haystack. If you look at the 3 billion base pair that we talked about, on average, what differentiates you with your neighbor or me, it's only 3 million positions. So it's only about 1 to 1,000. So this is a very small fraction, and what differentiates the normal cell to a cancer cell, it's only 10 to 1,000 positions. So we're talking about 10 to 1,000 positions that are different in your cancer cell compared to the 3 billion different positions that exist. So just as a scale, when you sequence your genome, it amounts to about 500 gigabytes of data, only once. So there are many different questions that we want to answer with data science. The first one is, how do you get cancer? How does it emerge? What are the risk factors? So now we can tell you that smoking is bad for you, but in the 60s, everyone was like, wait, yeah, smoking is good, you should do it, it's cool. What are the drivers? Meaning, what is the difference between a cancer cell? What changes it so that you get cancer, and what differentiates it from its neighbor that is non-cancerous? And then you want to predict things, such as what are the odds that you will survive from the treatment, survive from your cancer, and usually we use the metric of 5-year survival. And the bad news is sometimes you get a relapse. So what are the odds that you get a relapse from your first tumor? This is the part where usually it should be animated. Yes, cool. What you get on the left, so here it's just cells that are dividing. So 1 cell makes 2 cells, then 4 cells, and it goes on and on and on. But every time it divides, it accumulates changes. Usually you get 5 mutations per cell division. It's not very interesting. If you take an organ, you just see it's a bunch of cells. I don't understand what is the common origin of each one of them. And this is what is represented on the on the tree that is being built at the same time as the division. And this was the main question during my PhD: how do you can get the tree from just the picture that you get at one time? To rephrase it, so in yellow it should be normal cells, in orange it's cancer cells, and some of them have different logos. So some have pentagons, some have crosses, and it's just a change that they have got at one point, and that just emerges. So if you sequence, if you get the DNA of your cell, what you will measure is on the right, and you will say, well, there is a higher frequency of pentagons and a lesser frequency of crosses. The question is, how do you get the picture on the reverse? And this is what we did. We did something that was called quantum clone. It's built on expectation maximization, which is not very obviously used in Kaggle because it's usually you have to build it yourself. So what are the events? Usually you get gains and losses of chromosomes, you can get small mutations, and you have many observations that are changing what you are measuring, such as sampling effect, because you do not sample 10,000 times, you cannot sample 1 billion cells, you only have a few fraction of cells. So, how do you do? What is expectation maximization clustering? Well, you start with a random point for your clusters, and you just say, I don't know which point belongs to which cluster. So this is the initialization phase. Then you measure. This is the expectation. And you say, well, some cells are, or some points are more likely to belong to one of the two clusters. Then you update the position of your clusters. This is basically what we do with k-means, but k-means is definitive and does not give probabilities to belong to one of the two clusters. And in the end, when it doesn't move anymore, you have what is the final stage, and you just say, I'm stopping at this stage. So what you want to do is that you want to test this clustering technique for different numbers of clusters and then select them. So it's a huge amount of tests to do, and you have to find the best fitting method. So this relies on information theory and Bayesian information. So it's a lot of things just to give one graph, and this was the end of the paper, basically. This was our conclusion. 3 years of work just to say that, well, first you get cells that are very aggressive. They get a lot of mutations, so they are very sensitive to treatment because they cannot adapt anymore, because they are just growing so fast that they cannot do anything but growing. But some of them are okay. Well, it's still cancer, but they are okay. And when you treat them, they just don't die. Then, well, after a few years, these cells still had the trigger of cancer in them, and they just relapse. So this was the highlight of the paper, saying that this was a model that we could explain, and for the first time we tried to make sense of cancer using data science and how it behaved after treatment. So what is the conclusion of that in terms of data science? Well, in 3 years, what we spent time on was only the modelization part. We had the question that was only, how can you understand cancer? And that's it. My PhD was 1 sentence long, was what is clonal evolution in neuroblastoma? So what I spent 3 years on was only algorithm development, modelization, and packaging. So so far, when I say packaging, it's only a GitHub repo that it can be downloaded. It's not very nice if you have to interact with people that do not know anything about the field. And yes, this is one of the main drawbacks of academia, because you are only talking to people that are either from biology or data science. They just understand whatever you tell them because it's their field, and you do not interact with people that do not know anything about data science. So the thing is, I wanted to move from academia and get a so-called real job, according to my parents. And this is why I went to Kaggle. So thanks, Kaggle, for helping me get a job. And I started doing competitions that were close to my field, and usually we're talking about diseases. So in 2018, I don't know if you've noticed, but people developed a neural network that was able to predict if it's a melanoma or not, only from pictures, and physicians were getting a bit scared because they were thinking that their job is going all the way around, and it's not useful anymore. Truth is, it's not. And yes, for the first time, physicians were threatened by AI. So why did I participate? I first participated in Dream Challenge, which is another platform that only deals with academic research, and you get the paper instead of getting money, if you're interested. And it was to classify mutations. The other two, I participated, one by myself and one with our team of Kagglers at Converteo, which are them. They couldn't get here today, so I'm at least acknowledging them. They are all wonderful people, and you should get to see them. So we have taken 2 challenges since July. One was on pneumonia, which was X-ray detection, and the other one was center value prediction with a leakage and with a lot of questions with it. We'll get to it in a minute. So what can we learn from images, and what have we tried, what didn't work, what worked? The first thing that we did was to look at the data. And the thing is, we don't have a TPU, unfortunately. So we had only one GPU that wasn't so good. So when you get 1024 by 1024 images, you have to crop them and resize them. And once you look at them, you see that for a large amount of them, you have only a fraction of the picture that is useful. So we cropped everything that was fully black and not bringing in any information. Then we tried MRCNN because it was the one that was given as an example by Kaggle. And the thing is that it was a huge amount of data and computational burden just to train the data, and wasn't that useful because we only had boxes and not the segmentation image. So then we went YOLO, and not you only live once, but you only look once. And then we tried to predict the instances and regions. And truth is, it didn't work so well either. And in fact, for everyone, the loss was not very good at the end of the competition. Yes, we had a big issue, as Jean-François said, with our test set, and we were just overconfident in our training and in our predictions. So we went another way around, and we said, okay, we have the pictures, we also have patient information, can we add them, and can we stack them with one another? The thing is that we abandoned that very quickly because it did not work as expected, and it worked even poorly, and was even poorer than just taking YOLO by itself. And then we looked at what did the winners, and if you look at it, they just use stacking everywhere. It's like they trained 5 different neural networks with 5 different architectures, just blended everything. It was a nightmare, and when you have only one GPU, it cannot be done. So what's the big difference between academia and Kaggle? Well, if you are looking at the question, well, the question is already asked. You have the metric, you have everything, you have the data set, you do not have anything to collect. It's here, it just waits for you. And what you spend your time is modelization, being preparing your data, doing analysis, EDA, etc. But yes, some work is done by people that are not us, mainly the Kaggle team that creates the metric, that tries to understand the business value of the competition. And we have only a very short amount of time to do that, namely 1 or 2 months. So we are not developing new algorithm. You do not create a new neural network. You are just using and fine-tuning architectures that are already there. So this is the main difference between academia. When you have 3 years, you can just develop your own model. And to be honest, here it's just computational power is just going over the limit. If you have one image that is 124 by 124, what you are doing is that you are multiplying it by 3, because usually neural networks have 3 channels that are green, blue, and red. So here you are just using memory for nothing. And then comes the human behavior. So here comes internet. What are we trying to do? So where we are working is basically client knowledge, what you are going to buy, what you want to buy, and how much you are willing to pay for it. And then there is the thing that, how can I target you? Are you receiving emails? Are you likely to receive a call? Is it something just a pain if I call you, if I text you, or whatever? So what is the best channel to meet you? So these are the recent topics that we dealt with different partners. We dealt with not only machine learning topics. It can be visualization, even if sometimes it's just to make pretty graphics. Sometimes it's useful because you are talking to people that are not always fond of machine learning and that are not always as knowledgeable as you are. And then you have trying to score people and trying to score the probability that they buy your product. So, how does it work when you get a client? Usually, well, sometimes and more often, the question is, I need machine learning. And this is where you should be very vigilant. And this is also where I want to include you. Because if people just come at you and say, well, I want a machine learning model, just say no. Because there is a very high expectation right now in the artificial intelligence and machine learning field. And the issue is that it will get back to us at one point, and it will be really an issue in the next few years. So ask them what really is their question. What do they need prediction for? What do they need to understand from their data? What do they need to model? And there is a huge trade-off between the accuracy of a given model and the explication that can be taken from it. Even if we have seen this morning that a lot more work is being done for interpretability, it's still difficult to explain to someone that is not from the field what is a gradient boosted tree. There is a lot of questions that are listed here, but the final one is also of huge importance for your teams, for your clients, for your managers, is how long will it take for you to improve the model, given that you are often limited in time, in resources, and the efforts that we make on Kaggle for winning 0.00001 RMSE and 50 places is not always that useful for the business that is just behind and that just doesn't start with anything. Truth is, most of the time they don't have anything, and just getting a model to them is enough. So often, clients prefer to have simpler models with fine-tuned features and that get some insight to their problem and that they can activate instead of neural networks, which is a lot more fashionable. And as an example to what I'm saying is this was the final leaderboard for a sentence of value prediction challenge. So we were there with the Converteo team, and the thing is that you can see that the prize money here had such a small difference that it would make very little difference for the business value of it. So the final key point on that is, what is the difference between all of three? It's what makes the difference as a consultant when you only have 2 months and being in a competition when you have 2 months. The first thing is that we spend a lot of time just discussing with the client what is really that they need. We cannot spend a lot of time developing new algorithms. This is R&D and this isn't our work. But then what we used to do and what we are trained to is to develop and to produce an algorithm that can be used and that people can understand, which is a lot of work that is very different from academia. So the key factor here is to understand what the client wants and to define with him what are the metrics that they are going to use. Then deployment. We were talking this morning that proof of concept were often the case, and very few of them were pushed to mass produce, and very few were implemented. Here, this is what we focus on because this is what we want to give to our clients. And finally, the training, because not everyone is a Kaggler, and hopefully for you, because otherwise we wouldn't have so much job. But still, it's a lot of work to explain what you were trying to do and what you want to do to someone that is not as knowledgeable as you are. On that, I would like to thank you all for your patience and your attention. These are all the people that I've been working with over the past few years, either at Institut Curie, which is a cancer research center that you should log for if you didn't know, and also at Converteo and at Kaggle Days for the very nice organization. On a side note, well, we are 100 plus and we're looking for you. Thank you very much.
Hello. Thank you very much. Any questions? Okay.
I see you use Cat Boost.
Yes.
Why?
To be honest, I wasn't on that specific mission. We have some, well, we tried what works best in a limited time. So we tried both XGBoost like GBM and Cat Boost with more or less standard and default parameters. On that one, Cat Boost worked the best. So, and it was easily usable on virtual machines, which is another issue that we can find sometimes because we are not working on our own services. We are working on the client's one.